Next, perhaps I will go through some of these figures plotted in the study. So when I talk about how these individual studies cover diversity, this is really an outlier. These three papers from the beetles, you can see this, how evenly the coverage covered this megadiverse order. So firstly, let's go through how this figure was plotted. And now I've already showed you we've simply created a NUIC format file for the catalog of life taxonomy. So this component here is in simple NUIC format, and it's just plotted as that, just with some collapsed nodes. So what how I plotted this is to then go through each of the NUIC files and match the species IDs in these individual studies to this list of species in here, and get some positions along the X and Y axis too. So these are corresponding, each of these triangles is positioned corresponding to the equivalent species which has been matched with the taxonomy here. So here's the commands I used for plotting these figures. So this is a plotting tool I use for plotting the taxonomy, and you can see the input tree is what we've just produced, one of the NUIC format files for the catalog of life taxonomy for the beetles. So the main thing, let's look at this script, plot terminal points. That's those set of triangles corresponding to the matches. So several inputs from this. So one of the key ones is a set of y-axis values for a set of species. So you can see this set of beetle species and a single value. This corresponds to a plot. So effectively this is of course, the y-axis here. And during plotting this, it outputs, although the species aren't plotted onto the figure, they are read in, and the y-axis of each position is output along with the species, which is what we have here. So then we try and match these species names in each database phylogeny. So in beetles, there's about 200 database phylogenies, and they should be listed in this file here. This is the control file. And indeed, you can see a set of variables for the plot, and also the source tree list, just a space separated list of NUIC files. So these are the 200 beetle phylogenies database as part of the compilation. So you can see that we have source tree list here. So we are reading everything after the source tree list, which are put into an array going through each source tree. So we can stop this after the first source tree and see what it's doing. So we are then opening that file and pulling out all the binomials here. So let's print, we can simply print all the binomials found in Botzak et al. So what we are doing is trying to match that to the list of binomials, which are the y-axis values are stored, and the match for each binomial is made here. And if there is a match, then we store the set of y values into this hash here. So those are stored for the given source tree and then plotted along a sequence from left to right. So that's how this plot was created. So roughly depicting the species coverage for each of these